Hello, everyone, and welcome to another very exciting seminar in our series. My name is Julia Daroti. I'm a postdoc in GWAS lab at the University of British Columbia. And before I introduce today's speaker, I just want to remind everyone that, as always, there will be a Q&A session after the talk. If you would like to post questions for the speaker, you can do so in our Slack channel, where you can also upvote with a thumbs up the questions that you would like to see answered. Today, it is a pleasure to have with us Daniel Matute, assistant professor in the biology department at the University of North Carolina. Daniel obtained his degree in biology and microbiology at Universidad de los Andes in Colombia. He then completed his PhD in the lab of Jerry Coyne at the University of Chicago, working on the genetic architecture of reproductive isolation in different Drosophila species. He was then a postdoctoral researcher with Molly Szyborski, also at the University of Chicago, where he studied gene exchange between species and the evolution of reproductive isolation via reinforcement. He then moved to the University of North Carolina, where he is now, to start his own lab. His and his lab's work continues to address the question of how do species form and remain distinct, with a focus on understanding the genetic mechanisms and evolutionary consequences of traits involved in reproductive isolation. For his research, Daniel has received several awards, including the Dobzhansky Prize from the Society for the Study of Evolution. And today, he's going to tell us more about the evolution of postzygotic isolation during divergence. So thank you very much, Daniel, for accepting our invitation, and over to you.
Thank you, Julia, for that introduction, and thank you for all the organizers for not only bringing me here, but actually hosting this seminar series, which has been kind of a connection for many of us that are still at home. Today, I want to talk a little bit about some of the work that we have been doing, studying the genetic basis and the evolutionary processes that drive the formation of new species. And in particular, I want to talk about postzygotic isolation. When we actually talk about the formation of new species, we really are talking about how one species splits into two and starts accumulating genetic differences over time. The split might actually happen because of geography, might happen because of ecological differences. But what really matters here is that over time, the resulting splits are gonna become different from each other. How different? That is essentially the question that the field of speciation tries to address. And is we try to understand what happens when, or what is gonna happen when those nascent populations actually come into secondary contact, when they have the possibility of encountering each other and producing progeny. There are several possibilities, of course, and the first one is that the mechanisms or the barriers that actually have, like the differences that have been accumulating over time, keeping these species apart, are just not strong enough. And in that case, there is going to be a collapse of what was a speciation process in motion. The other possibility is that those two nascent populations actually persist, even in the homogenizing phase of gene flow. In that case, the process of speciation has happened. In that regard, it's actually quite important to try to understand what phenotypes keep these species apart. And to that end, there has been many classifications of these type of traits. And the ones that I actually try to use essentially when I am thinking about the process are the ones that actually are regarding reproductive stages in the life of an organism. You can actually describe the reproductive life of an organism as two big events. The first one is essentially mating, and the second one is zygote formation. So if a barrier, if a phenotype actually precludes the possibility of gene flow, and before mating takes place, then we actually call that barrier pre-mating, and includes behavioral traits, it includes habitat isolation, and even includes geography. If the barrier or the phenotype actually takes place after mating has taken place, but before a zygote gets formed, then we call those barriers post-mating prezygotic, and includes every trait that essentially involves the interaction between gametes or between female reproductive tracts and sperm, for example. Now, if those barriers occur before the zygote gets formed, after the zygote gets formed, we essentially call those barriers postzygotic. In this regard, this classification of prezygotic, postzygotic, pre-mating, post-mating has been in place since the 1930s when Dobzhansky published his famous book in 1937. But there are several papers that have been seminal to actually how we understand the process of species formation. And one of them was this paper by Jerry Coyne and Allen Orr in 1989. I didn't read this paper when it came out because I was in third grade, but I read it essentially the second day that I started doing my PhD, and the paper has seminal influences on essentially what we do right now in speciation. And one of them is that by compiling data on the magnitude of reproductive isolation of Drosophila and using the phylogenetic method to try to assess how divergence essentially affected the magnitude of reproductive isolation, they came up to the realization that in general, in Drosophila, reproductive isolation follows an increase over time. The more diverged that two species are, the more likely they are to be isolated. This is, of course, not absolute, and there are exceptions, but in general, there is this trend. But essentially, the first really novel insight that this paper provided is that in Drosophila, pre-mating isolation tends to evolve or accumulate faster than postzygotic isolation. This led to essentially the mantra of the field that pre-mating isolation is more important for speciation than postzygotic isolation. We have actually repeated this experiment several times as a field, including in my own lab. In 2018, David Turrisini published a paper that was similar to Cohen and Orr, but actually trying to study the evolution of post-mating pre-zygotic isolation in Drosophila species. And we essentially come to a similar conclusion to Cohen and Orr, and is that behavioral isolation tends to accumulate much faster than postzygotic isolation. But post-mating pre-zygotic phenomenon tend to evolve also quite fast, almost similar to behavioral isolation. This is the point when Brandon Cooper and I decided to gather all the possible information that we could by reviewing the literature and find out whether the statement that pre-mating isolation tends to evolve faster than postzygotic isolation is really true. And we find support for this assertion in several clades. For example, in darters, in sticklebacks, and in several plants like sexually deceptive orchids and streptanthus. But we also find that there are other clades in which this assertion is just not true. There are several cases in which pre-mating isolation accumulates about the same rate or even slower than postzygotic isolation. So the idea that pre-mating isolation is always faster and always more important than postzygotic isolation just didn't ring true. This, coupled with the work that Jen Coughlin has done in my lab, showing that there is a monotonic increase in the magnitude of postzygotic isolation in pretty much any clade that we actually look at. And the way that she did was essentially just to review the literature and just do the same thing that Cohen and Orr did, essentially regress the magnitude of genetic diversity, of genetic divergence on the x-axis, and the magnitude of postzygotic isolation in the y. And you see that in all cases, ranging from birds to several types of plants, there is an increase on postzygotic isolation. I wouldn't go as far as actually telling you that in different clades, there is differences at the rate at which postzygotic isolation accumulates, simply because the metrics, and this is an important limitation of this data collection, is that the metrics of genetic divergence are actually quite different among different taxa, as you can see as specified in the x-axis. But what actually we can know is that there is an increase, regardless of essentially which way you slice it. So this importance of postzygotic isolation, this monotonic increase, and this idea that postzygotic isolation sometimes evolves quite fast, essentially just led us to revisit some of the seminal conclusions of Cohen and Orr as a seminal paper of the field. And one of them is that pre-mating isolation tends to be more important for speciation, especially in sympatry. This is the representation that essentially they show by collecting essentially compiling the Drosophila data and split them in allopatric and sympatric pairs. They essentially concluded that the mean pre-mating isolation tends to be much stronger in sympatry than in allopatry. But that was not the case for post-mating isolation. This essentially was interpreted along with some seminal cases of a study of stronger reproductive isolation in sympatry by Lierman and Terry Markow, that reinforcement was a pervasive force in nature. And since then, we have actually provided several, as a field, we have provided several examples that show that reinforcement might actually be quite common in nature. But we wanted to assess whether this idea that pre-mating isolation is the only trait that accumulates faster in sympatry was really true or not. And for this, we were expecting, essentially, if the results from from this paper actually we could recapitulate them, we would expect that pre-mating isolation in sympatry would evolve much faster than in allopatry, and that postzygotic isolation would show about the same similar rates of evolution or similar rates of accumulation. And for this, we wouldn't have been able to do it without the excellent compilation that Roman Jukilevich published in 2012 in Evolution, which was essentially moved the original collection of data from 190 species to over 600 species of Drosophila, which essentially gave us a fair bit of more power to try to assess whether the rate of pre-mating and postzygotic isolation is similar in sympatry and allopatry. So the first thing that we did was to run a sanity check and was try to determine whether pre-mating isolation truly evolves faster in sympatry than in allopatry. And again, we did this with Brandon Cooper. And unlike several approaches, we actually use a logistic regression instead of a linear regression. This is essentially based on multiple modeling papers, like, for example, by Tammy Melson and James Mallet and Dan Polnick, that actually have shown that there is a lag on the accumulation of reproductive isolation to a rapid increase and then an asymptote, because reproductive isolation cannot be higher than complete. And indeed, we find that pre-mating isolation evolves faster in sympatry in the way that it reaches this inflection point much faster in sympatry than in allopatry, and it also reaches the asymptote much faster. And a different way to actually represent these results is actually just look at the histograms of several bootstrap distributions of the inflection point for pre-mating isolation. And we find that indeed the inflection point is much lower for sympatry than for allopatry, confirming the idea that pre-mating isolation does accumulate faster in sympatry. With this sanity check already in mind, we essentially move to the question of whether post-mating isolation actually show a similar pattern to sympatry to pre-mating isolation, or whether actually it replicated the results of Cohen and Orr. And the results actually were, at the beginning, a little bit surprising, in the sense that what we find that postzygotic isolation also evolves much faster in sympatry in Drosophila than it occurs in allopatry, as it is shown here, by the faster asymptotic, reaching the asymptote, but also by the much lower inflection point than the sympatric lines show with allopathic lines. When we essentially just expand these studies to look at not only Drosophila, but Lepidopterans and Buffotoads, we find similar patterns. The inflection points are much lower for both for Lepidopterans and to a lesser extent in Buffotoads, showing, or at least suggesting, that postzygotic isolation also accumulates faster in sympatry. This essentially just throws a wrench at the conclusion that reinforcing of pre-mating isolation is the force driving this stronger reproductive isolation in sympatry. And at the beginning, this caught us by surprise. But when we started going deeper into the literature, we found that this report has been to some extent found before. Dean Castillo has a paper in 2017 that actually reports something very similar. So we went on to actually trying to figure out what were the possible explanations for a pattern like this in sympatry. That essentially, and the pattern is that pre-mating and postzygotic isolation both accumulate faster in sympatry than in allopatry. The first possibility is, of course, reinforcement, but not only of the pre-mating type, but also of the post-zygotic type. This is an idea that has been put forward in the literature before. Wallace, in his book Darwinism, actually posits forward in the chapter 7 when he talks about hybridization. And Jerry Coyne himself in 1974, his first graduate school paper was about the possibility of reinforcement at the postzygotic level. There is a complication in Drosophila in particular, and is that Drosophila has internal fertilization but external development, so it's unclear what would be the advantage of reinforcing postzygotic isolation. The second possibility is differential fusion, also known as the Templeton effect. This is another possibility that was put forward over 100 years ago by Wallace, also in chapter 7, and by Alan Templeton and formalized to some extent by Alan Templeton in 1981. And the idea involves that when you have a species in allopatry that are polymorphic for their levels of reproductive isolation, shown here by the different shades of the circles, when those species and dark circles being strong reproductive isolation, soft color circles being weak reproductive isolation, what you can expect is that if these species come into secondary contact, those individuals that have weak reproductive isolation are simply gonna go extinct. And are gonna go extinct because of hybridization itself. This is an idea that was just essentially discounted and put to the side in the field because of the result that postzygotic isolation was simply not stronger in sympatry as exemplified by the results of Jerry Coyne and Alan Orr. But with the addition of data that Roman Jukiewicz compilation allows us to do, we're actually finding support for this hypothesis. So the question, when I try to rephrase the question of whether is pre-mating the only mechanism or the only barrier that is stronger in sympatry, the answer is a categoric no. But the answer of why this is the case is actually a lot murkier. And is that I cannot really tell you whether differential fusion is actually occurring here or not, because there are no formal tests for the idea. But what is clear is that reinforcement of pre-zygotic isolation, of pre-mating isolation, is not a suitable explanation for the patterns of a stronger reproductive isolation in sympatry. And as I told you before, the process of divergence essentially just includes accumulating and increasing reproductive isolation over time. So I want to change gears a little bit and talk about not only what happens in general patterns in nature when you compare multiple species, but also try to actually talk a little bit about what are the genetic underpinnings of what happens in postzygotic isolation. But I think that there is an important disclaimer to put forward, and is that not all reproductive isolation is the same. If, for example, you are studying species pairs that are of low divergence, and there is just a few cases of reproductive isolating barriers that stop gene flow, those cases of species are actually quite good to study how a species form in the first place. What is the first changes that actually lead to species formation? Once you start accruing more divergence, or when the species start accruing more divergence and the number of barriers increases, those are not so good to study what was the first trait that actually changed, if that ever happens. But actually, when there are multiple barriers actually working together or working sounds deterministic, but that's not the case. When they're actually stopping gene together, those are good species pairs to understand essentially how a species persists in the face of gene flow. Now, when you have really divergent or highly divergent species, and there are plenty of barriers that actually separate the two species, those are not really very good to study speciation itself, because you cannot simply identify what were the first barriers to appear and which ones are epiphenomena that just occur because of divergence. In this particular case, though, there is a lot of information to be surmised. And for example, you can find out how much genome divergence occurs as evolution is proceeding, or how much genomic divergence actually leads to the separation of a species over time after speciation has occurred. And I actually want to give context on why, one of the reasons, some reasons why we study Drosophila. First, it's a very specious group. There is over 1,600 species and maybe many more. Here I'm showing just an example of how many species there are. Essentially, this is 155 genome phylogenomics approach that we use to reconstruct some of the relationships of the genus Drosophila. This was done in collaboration with Adnan Soborov, Bernard Kim, and Aaron Komol, who actually did the vast majority of the work. And in this case, what we did was to generate new reference genomes to try to find out what were the genetic relationships of the gene. We generated de novo reference genomes to find out what were the genetic relationships of the species in the genus. And we resolved the phylogeny, or at least the phylogenetic relationships of the species that we were able to include. And confirming some previous approaches, we realized, or we revealed that the group of the Drosophila group is very old. It's on the order of 50 million years old. But a phylogeny by itself doesn't really tell us much. The second reason why this group is important is because introgression seems to be common. And again, in the same group of excellent people that I have been working with, has been able to actually use multiple approaches and try to determine how much gene flow occurs between these species. Here I am just showing an example of one of the variations, the melanogaster species group. And each square in the triangular matrix implies that we did triad comparisons and try to find out whether there is introgression by looking at multiple genes, multiple methods to detect gene flow. And those squares that are purple show instances in which we were not able to detect gene flow, but those that are orange actually reveal cases in which there is evidence for gene exchange. Some of them happen between like there is the evidence of admixture between quite divergent species, which tell us, and if you would try to essentially reconstruct the path in which these species hybridize, that actually introgression is actually quite old, and that what we are seeing in these phylogenetic trees is the remnant of really old admixture. So there is a second reason. This is a second reason to study reproductive isolation in Drosophila, because introgression seems to occur. So it's important to try to find out what are the traits that actually keep these species apart. And the third reason is that different traits accumulate at different rates. Different traits in particular of postzygotic isolation. This study led by David Turrisini essentially found out what was the median age at which female fertility, male fertility, and male viability essentially started breaking apart in Drosophila hybrids. So in the x-axis, you have the magnitude of genetic divergence, and in the y-axis, you have essentially the density of the function. About 5% of genetic distance, you can almost be certain that in Drosophila at least, you have 5% of genomic divergence that your males are going to be sterile. If you move further, about 10%, you're going to find females that are sterile, and about 15%, you're going to find males that are inviable. Please note that the female viability is not represented here because we just simply didn't have enough data to conclude when it would happen, but it seems to happen after 20% of divergence, which is quite high. So let me just go back to this table. And for the day, I am going to just talk about cases of postzygotic isolation when there is really high divergence. And this is because of two reasons. First, there is excellent examples that have been published recently about the genetic basis of reproductive isolation in closely related species. These four examples that I am showing here, from Siphophorus to bees to Mimulus to Drosophila, are just papers that have appeared in 2020, and there is only 8 months, like 9 months so far. So this is a field that is definitely exploding with absolutely excellent research. And the second reason is because even in Drosophila, we have actually studied the genetic basis of hybrid breakdown in hybrids, but to some extent, we have been limited to study up to 5 million years apart. Hybrids between melanogaster and chimeras have been a workhorse of the field and have revealed important insights in the field since the 1920s when they were reported for the first time. But there are hybrids that actually can be produced at much deeper levels of divergence. Just about 10 years ago, we found out that we could hybridize melanogaster with Xanthomea, that is a species that is endemic to the Gulf of Guinea. This essentially will go down as just another hybrid that is produced in the Drosophila phylogeny. It wouldn't have much impact. But we were quite curious about what were the phenotypes that actually cause reproductive isolation in these hybrids. Namely, when we look at the embryos, when the cross only produces females that are sterile. Adult females that are sterile. The adult males are absent. So we essentially just try to figure out, do a survey of the development of these hybrid males, and we find that they die as embryos. When we open these embryos, we find that they have horrific developmental defects. So if, for example, you compare two individuals that are wild type, melanogaster and Xanthomea, you see that this is the head, this is the telson, so the tail, and you have abdominal segments that are in essentially these bands that you see right here. You see about the same thing in Xanthomea. Essentially, the phenotype is the same. When you produce F1 females between these two species, you see essentially the same pattern with some defects in the banding pattern, but nothing too pronounced. But when you look at the males, essentially you see a pattern that I had never seen before. And that is that you have a head, you have a telson, and you are missing complete abdominal segments, which turns out to be quite lethal. So we went on to the quest of trying to find out what was the genetic basis of this particular trait. And one way to do it is essentially we use a mixed approach in the sense that we use the Drosophila genetics reference panel, that is a set of inbred lines that have been produced over, like, essentially just doing brother mating systems for multiple generations and crossing them to Xanthomea and actually measuring the frequency of hybrid viability and of the abdominal ablation. And what we see is essentially what I am showing here. In the upper panel, you have a Manhattan plot for hybrid viability, and in the lower panel, you have a Manhattan plot for the rate of abdominal ablation. And you see that the rates of abdominal ablation and hybrid viability are concordant, and both of them are caused by a segment of the tip of the X chromosome here. But before I tell you what is under this peak, let me just give you a little bit of context on how we have been working on this before. Drosophila genetics actually allows you to do several tricks, and one of them was to try to introduce small pieces of the X chromosome from melanogaster into hybrids and see essentially how they fare. And by introducing small pieces, we could just style the whole X chromosome and determine whether those small pieces cause hybrid inviability or not. Indeed, there are several pieces that cause hybrid inviability that are shown here by the solid or the striped colors. There are others that just don't have any perceivable effect. But in the case of this segment that I am showing here, that is in the cytological band 3, is concordant with the GWAS results. And moreover, this is the only band, this is the only segment of chromosome that causes these horrific abdominal ablations. So by just using a few more tools of Drosophila genetics, including a few more duplications of the X chromosome, which I would be happy to discuss later, we find that the only allele that resides in this region is an allele called giant. It's a gene called giant. Giant is actually fundamental for early Drosophila development. So here I am showing some of the early studies on Drosophila development that show that when you have giant mutants, you see defects on the formation of abdominal segments that are essentially completely absent in the lower part of this panel here. Moreover, there is exquisite surveys of gene expression on genes involved in Drosophila development. In the case of giant, we know that it has an anterior expression, and we know that it has a posterior expression that is concordant with the abdominal segments that are lost in melanogaster xanthomea hybrids. This is essentially another way to see it. When you do cuticles, or essentially you extract the dead larvae from the embryos. In Drosophila mutants, you see these defects of A6 and A7 that are abdominal segments that are completely just malformed here, and that has consequences for the downstream gene expression, as you see here by these stainings. And it's notable that when we actually do cuticles of those hybrid embryos, we see those are the segments that are missing. Some of the segments that are missing in particular. So we did just a genetic test, and we essentially tried to find out whether when we took null alleles of giant, essentially just a completely misformed protein that forms this allele giant X11, we tried to see whether they produce ablated embryos. So let me just walk you through this slide. In the x-axis, you have the genotype, and in the y-axis, you have the proportion of ablated embryos. This is essentially the frequency of ablated embryos in females, and this is the frequency of ablated embryos in males. You see that is much higher. And when you actually have hybrids that don't have the giant melanogaster, the giant X11, and have the allele giant X11, they just simply don't show ablated embryos. They die, but they don't die because this abdominal defect. They die for other reasons. When we actually look at other segments of the X chromosome, we see that there is no reduction in the proportion of ablated embryos compared to other females, that essentially females that have the two complete chromosomes that are shown here by this red and this blue. So this giant melanogaster allele is actually quite important to producing these defects in the abdomen in melanogaster xanthomea hybrids of this type. Which is, and in case that if you can see it, this is essentially the intestine connecting the head, the thorax, and the telson. That is the last bit of the abdomen. There is more, because Drosophila hybrids are actually quite powerful in the sense that we can produce several. And it happens that melanogaster is not the only species that can cross with xanthomea. We can actually cross simulans, mauritiana, and seychellia. And we can actually just survey whether these hybrids between simulans and xanthomea, mauritiana and xanthomea, and seychellia and xanthomea also have abdominal issues. And this essentially reveals two things. It reveals, well, one thing with two possible outcomes. It reveals whether the allele from giant melanogaster evolved after simulans and melanogaster split apart, or whether it's common to all these species, and thus is suggestive of an evolution of that particular change of what we call giant melanogaster causing hybrid inviability before these species split apart. So older than 5 million years ago. So when we look at melanogaster xanthomea hybrids, as I show you, they have this defect. But when we look at melanogaster simulans, and the others, melanogaster xanthomeas are hybrids, we see the same defect, indicating that there is a common genetic allele to Simulus, Mauritiana, Seychellia, and melanogaster that actually causes that inviability with xanthomea. So this is a very old genetic change. But there is, of course, more that we can do. Allen Orr in 1993 reported that you could actually hybridize Simulus with Tessieri, and this essentially just gave us a lead to try to find out what would happen if we cross all these species to Tessieri and we look at the abdominal, their abdominal segments. And when we do a survey of this time, we see that that the abdominal phenotype is simply not present in melanogaster Tessieri hybrids. As a matter of fact, there is almost no embryonic death in these hybrids, which actually tells us something else here, and is that, okay, we figure out that the giant melanogaster is not exclusive to melanogaster, that also is shared between Simulus, Mauritiana, and Seychellia, and evolved between 10 to 15 and 3 million years apart in this branch. But the fact that actually Tessieri, when hybridized with melanogaster, doesn't show an abdominal defect tells us that there is a set of allele or a set of alleles in the xanthomea genome that actually is private to the xanthomea and actually is not shared with Tessieri, indicating that there have been changes that actually have occurred in this branch after these two species split apart. This result essentially tells us that the evolution of hybrid inviability in this species between melanogaster and xanthomea had absolutely nothing to do with the speciation, and that hybrid inviability is just essentially a phenomenon of multiple changes in multiple stages in the branching pattern of the evolutionary tree of this species. Working with Heidi Mavinger, Jeremy Wang, and David Pitt, we have been able to produce xanthomea assemblies, de novo assemblies, that to try to understand the magnitude of genetic variation in xanthomea and other species as well. And we have found that there is just no evidence for positive selection in giant or in tailless. As you can see, I'm here showing just the results of PAML, but you can actually do McDonald-Kryman test, and essentially you can actually do any analysis of selection and you will find no selection. Which is actually more informative, is that if you were to compare giant to the rest of the genome, giant actually falls in the bottom quarter in terms of rates of evolution, rates of change. So essentially it's a gene that is evolving quite slowly. But there are particularities in the melanogaster and xanthomea hybrid, melanogaster and xanthomea giant alleles. One of them that is showing here is actually that the coding sequence of giant actually differs around 500 base pairs. And what is in this region is a region of low complexity that has a long stretch of polyQs. We don't really know whether this stretch of polyQs actually is involved in hybrid incompatibility, but we know that is one of the notable differences between melanogaster and xanthomea giant alleles. Nonetheless, these changes in coding sequences doesn't really tell us whether the coding sequence actually matters for causing hybrid inviability, or whether there is non-coding DNA that is actually causing the hybrid incompatibility. And this is essentially where the work of Wen-Han Chang at the University of Chicago comes into play, because he had the idea of generating chimeras for giant that included the non-coding region of xanthomea and the coding region of melanogaster, and the reciprocal as well, find out using CRISPR. So essentially what he did was also to generate non-coding region for each of the two species and mix them with the coding region of the other one. This allowed us to do a couple of tests. One of them was to test what happens when you have in an F1 genetic background, in a hybrid background, what happens when you have the non-coding region from xanthomea and the coding region from melanogaster. The second test that we could do was what would happen if you have an F1 background and you have the coding region from xanthomea and the non-coding region from melanogaster. So let me walk you through this. Essentially, what you have here in the x-axis is the four possible genotypes of the chimeras, including the ones that match the species, from the same species, melanogaster, and xanthomea, and the relative viability to essentially we call wild-type hybrids that have essentially no mutations. In the case of the chimera that has melanogaster non-coding regions and the coding region from melanogaster as well, we see a decreasing viability that is significant compared to wild-type hybrids. Notably, we see the same when we have a non-coding region from xanthomea and a coding region from melanogaster. We see also a much lower viability compared to wild-type hybrids. When we have a non-coding region from melanogaster driving the expression of a coding region from xanthomea, we see no difference from the relative viability from wild-type F1 hybrid. And when we have the two elements from xanthomea, the non-coding region and the coding region from xanthomea, we see an increase in viability. This latter result, I don't know what it means. And I would be happy to essentially take suggestions. But what I do on what the results that I can surmise from these experiments is that the CDS from melanogaster with being driven by either the non-coding region from melanogaster or from xanthomea causes a decrease in viability, which is telling me that at least the coding region of melanogaster is implicated in the phenomenon of hybrid inviability. This is a point in which I was quite curious, and I have been working with hybrid incompatibilities for a while, and we had identified giant as one of the parameters on hybrid inviability. But the very basics of epistasis is that you must have at least two partners that actually, when you have a hybrid incompatibility that just don't work well in the hybrid and just lead to hybrid breakdown. So I essentially was intrigued and tried to get my team to work on trying to find out what was the partner of giant melanogaster. And one of the ways that we did this was essentially take the same panel of almost isogenic lines of the DGRP and try to introgress the giant mutant, but that also meant that we had to introgress a chromosome that was a control, a balancer chromosome. We did this in over 200 different backgrounds. And the reason to do this was that if we actually were able to have these two chromosomes, the giant mutant, in different autosomal backgrounds, we could try to use essentially a genome-wide association mapping to try to find out what was the autosomal partners of giant that were interacting and essentially were causing hybrid inviability, provided this was limited insofar as we were using the variation within melanogaster, but we were hopeful about it. And indeed, what I am showing you here is the results for that for those introgressions, crossing to xanthomea and finding out whether there was any alleles in the genome of melanogaster that were associated with the penetrance of giant melanogaster causing hybrid inviability in these hybrids, both in female and in males. And what you see here is that there is a rather large peak on the third chromosome, almost at the end of the third chromosome. That peak has only 9 alleles that I am actually showing here. These 9 alleles, actually, of these 9 alleles, tailless, that I'm showing here in green, is the only one that is known to actually genetically interact with tailless. So we wanted to know how did that happen, whether actually this signal that we were getting from tailless was important. And the work that we were doing with tailless was not essentially a wild goose chase, because we knew from the excellent work that has been done in developmental genetics that giant and tailless interact genetically, and that tailless actually interacts with giant to actually coordinate flight development at the early embryo stage. For full disclosure, after we identified these 9 alleles, we only follow tailless, so there is a possibility that the other 8 are actually implicated, but I thought that it was a pretty good lead that we could follow on tailless. So what we did was to generate disruptions of the tailless allele in xanthomea and melanogaster, aided by rainbow transgenics. And this allowed us to do a couple of tests. The first one was what would happen in a hybrid background if you have a mutant allele for giant melanogaster. Remember, that's essentially one of the allele that causes these abdominal ablations, and also had a disrupted allele of tailless from xanthomea. But also allows us to test what would happen if we have a similar genetic background in which the only difference was that the disrupted allele was tailless from melanogaster. And I'm going to show you those results here. So in the x-axis, you have genotype, and the y-axis, you have relative viability. The blue line in this case is the fitness of what essentially we call a wild-type hybrid that has a functional giant melanogaster, a functional giant centomere, and functional allele of tails from the two species. In the y-axis, I am actually symbolizing whether an allele is wild-type by gray and whether it is mutated, essentially disrupted, by essentially writing it in black. So let's start with the first experiment. What happens if you have a tailless from melanogaster that is disrupted in an otherwise wild-type hybrid background? What you get is essentially a very modern increase in viability. It's not the largest, it's nothing that I would consider to write home about, but it does exist. That compared to the effect of actually disrupting giant melanogaster that I had already shown you before is actually quite minimal. It does exist, but it's quite minimal. So this is essentially the results that I have been showing you so far. If you disrupt giant melanogaster, the viability just increases significantly. If you disrupt the tailless from centomere, there is really no difference from the relative viability that you obtain in these hybrids. So disrupting the tailless from centomere has no effect. But what I was particularly intrigued was what would happen in cases in which you had the two essentially double mutants. When you have a giant melanogaster and a tailless melanogaster disrupted at the same time, you see that the viability actually increases quite a bit compared to the wild type. And when you have a giant melanogaster disrupted and a tailless centomere disrupted, you see that the viability of these hybrids is not significantly different from just hybrids that have the giant melanogaster disrupted. So essentially what this is telling us, or the way that we have interpreted these results, is that the effect of tailless melanogaster and giant melanogaster together actually works together to cause hybrid inviability. So the conclusions from these experiments, from this genetic survey that actually included finding out the genetic partner of giant using introgressions and GWAS, and following those results with reciprocal gene disruptions in the melanogaster and centomere background, what those results are telling is that giant melanogaster and tailless melanogaster interact with something in the centomere genome to cause hybrid inviability. And that's something we don't know still what it is. Emmanuel D'Agostino in my lab has actually working on generating multiple lines of centomere to do similar experiments. But to date, we don't have any idea what it could be. So essentially, that red branch that I showed you in the phylogeny earlier, that essentially evolved after centomere and the series split, we don't have any idea what that could be. But what I can tell you is that our data suggests that there have been at least two changes, giant melanogaster and tailless melanogaster, that actually have worked together, that have evolved in the melanogaster branch to cause hybrid inviability in these melanogaster centomere hybrids. So what I have shown you is that two alleles that are involved in a single hybrid incompatibility. You might actually be of the mind that it really doesn't matter what the identity of the genes is, and I understand that perspective. I mean, if it is not this gene, it must be another. But what these results are telling us, I think that is larger than just the identity of the genes, because what they are telling us is that hybrid incompatibility reveals that there is cryptic evolution at the molecular level in a way that was predicted by True and Haig in 2001, in the sense that giant melanogaster and tailless melanogaster are absolutely crucial for the body plan development of Drosophila, both in centomere and in melanogaster. And if you actually look at the phenotypes, they are quite similar. But the genetic underpinnings of these changes actually have changed, and it has occurred in an absolutely fundamental developmental plan. So if I were to revisit the original table that actually I told you that highly diverged hybrids are important to understand how genome divergence occurs, I would essentially just say that hybrids are one of the basic complementation tests that we can do, and hybrid viability is just a complementation test, just not working. Essentially, it's telling me that the allele from giant from melanogaster and the allele from giant from centomere are just not equivalent. Moreover, it indicates that there is developmental regulators that are co-evolving within the genomes, and that even though we see similar phenotypes, there is just divergence that we are not able to detect just by looking at sequence that are just undergoing in each of these branches of the phylogeny. if I were to revisit essentially the original question, and it's essentially the evolution of postzygotic isolation during divergence, what is the importance? I would essentially just take three important messages here. And this is essentially my final thoughts, and is that reproductive isolation as a phenotype is not the same as a speciation. It's actually quite difficult for me to wrap my head as what an speciation phenotype would be. But I can tell you that in the case of melanogaster xanthomea, even though there is hybrid compatibility, hybrid viability, that we can pinpoint at the molecular level, or at least at the genetic level, we know for a fact, not for a fact, our evidence is telling us that this reproductive isolation was completely detached from speciation processes. So it is important to actually keep this difference in mind. Nonetheless, postzygotic isolation is an important marker of divergence. And in cases in which species actually might form quite often, as was elegantly put by a paper by Rosenblum and Panel and Harmon, species might form all the time. But what actually might matter is not how often they form, but how often they persist, for multiple reasons. The species might actually go extinct because they get ecologically displaced, because they were extinct by hybridization, by competition. Like, there are multiple reasons for this. But at the end, what we see in terms of biodiversity are the species that persisted and not necessarily the ones that form. Finally, I would like to actually just say that even though we have multiple cases in which alleles that are involved in postzygotic isolation have strong marks of positive selection, as it is the case of nuclear pore proteins in Drosophila and HMR also in Drosophila, the case of giant is a little bit different and actually points out that there is no evidence of positive selection or any natural selection actually acting on the evolution of this allele. That doesn't mean that it hasn't happened somewhere else in the pathway, but it means that it is important to keep in mind that the phenomenon that we are seeing in hybrid viability between melanogaster and xanthomea might just be the epiphenomenon of just cascading changes that have occurred in the genome. I want to finalize thanking the people that have done the work. Brandon has been instrumental on the thoughts and the execution of the first part of the talk, thinking about postzygotic isolation as a general pattern and actually revisiting the ideas from Cohen and Orr now that it has been 30 years since the paper was published. And when Han-Chak has done exquisite molecular work trying to find out what are the genetic pinpoints, the genetic changes that actually have led Giant to cause hybrid incompatibility. These are other people that actually have been also involved in the project on the sides. And I want to actually thank all the people that have read these multiple iterations of the manuscripts that we have written about hybrid incompatibility, the funding sources, and announce that with Danish writer, we are actually recruiting postdocs. So if evolutionary processes is something that you're interested, please don't doubt on contacting us. Thank you very much. And I will be happy to take any questions, all the questions.
Thank you very much. That was very interesting. Let's see. We have a question asking, why do you think there are sex differences in the degree of genetic difference between Drosophila species that leads to sterility?
Okay, so let me just try to reframe the question. The question is, why do hybrid viability and hybrid sterility evolve at different times in Drosophila?
I think it's asking more about the sex differences in degree of genetic difference between Drosophila species that then leads to sterility. Maybe I'm...
Okay, I am going to do what one does, and I am going to answer the question that I think was asked, but I will happily be corrected. This is essentially, I think that the question addresses the phenomenon of Haldane's rule, and is why in cases in which there is differences in the magnitude of postzygotic isolation, in particular of intrinsic postzygotic isolation and developmentally based postzygotic isolation, there is differences between females and males. This might be caused by multiple reasons. One of them is just hemizygosity, but there is also a hypothesis on faster male effects. And essentially, this is a very large field of research on speciation. And I am not sure if I am addressing the question, though.
There's another question asking, could you comment on the relative importance you think inviability or infertility have versus more subtle reductions in fitness have in speciation?
Oh, absolutely. This is one of my favorite topics to discuss, because there are multiple reasons why we have actually focused on sterility and inviability. I don't doubt that there are very important. But I also don't doubt that one of the reasons why we focus as a field is because there are very strong phenotypes that we can recapitulate in the lab relatively easily. So, for example, there is excellent research that has been done in the last few years that exemplifies how, for example, how hybrid birds have problems learning or how they have problems migrating. These are, in all likelihood, they might be very important. And when you put hybrids in the field, they might actually just be even more important than other phenotypes that we observe late in divergence, right? In the case of inviability, you cannot have hybrid defects in cases that the hybrids are not alive, right? But I am certain that the reason why we don't know the rates of evolution of all these other defects that actually might be more subtle, and by subtle mean recapitulated in laboratory conditions, is because collecting them is quite hard. And those are experiments, those have to be large field experiments that have been done for multiple species in order to get power. So as a field, I think that it would be phenomenal to actually find out what they are, but I also acknowledge that it's actually quite difficult to pinpoint the rate of evolution and importance on the speciation of these defects.
Yeah, that would be very interesting to achieve that. There's another question asking, if reproductive isolation is not the same as speciation, then what happens with the biological concept of species?
Okay, this is a great question, and I would like to essentially just clarify what I mean that reproductive isolation is not the same as speciation. In the case, studying a phenotype that actually causes reproductive isolation, like hybrid inviability in Drosophila, is certainly not the same as actually finding out what were the causes of a speciation in Drosophila. That's what I meant. I think that the biological species concept has a place in the sense that it connects the formation of a species with a process, but I am not an absolutist about it. Like, half of my lab works in fungi, in which we cannot do crosses, in which we cannot simply dream of applying the biological species concept. So there are surrogates to it. So I think that one has to be a little bit flexible about what is the best tool to address the question that one has at hand.
Thank you for that. There's another question asking, do you think key early developmental genes like giant ever evolve fast enough to contribute to speciation?
Okay, so there is a phenomenal preprint by Peter Ruff on this. And essentially, they just take the concept of genetics of systems drift and just take it to a quantitative genetic model, trying to find out whether it causes speciation or not. I don't know whether I am ready to actually say that any particular, not only early development, but any particular biological process is leading to a speciation in multiple cases. We simply don't have that information. Like the identity of many, there is not that many alleles that are known to just really cause reproductive isolation. So drawing patterns from relatively small numbers, I am a little bit wary
of. Another question is, as an alternative explanation to the Templeton gap for elevated isolation given in sympatry given divergence, could introgression in sympatry make species more related but not impact isolation?
Yeah, absolutely. That's something that we have actually been toying around. And it's essentially a limitation that was on passing acknowledged by Cohen and Oren, is that in sympatry, you're going to have gene flow, right? Which is something that you're not going to have in allopatry by definition. And that might actually just drive the perceived genetic distance to essentially be lower in sympatric species and actually just essentially kind of bias all the effects that we have. The other possibility, of course, is that it's biological, and is that introgression just gets rid of a bunch of hybrid incompatibilities, but I don't really know which direction that would affect. But definitely introgression is something that has to be put in context in these comparative studies of reproductive isolation.
Thank you for that. Beyond the genetic consequences of postzygotic isolation in the species complex you study, what are the ecological consequences? These different species or splitting populations can coexist in relation with the resources they exploit?
That's a phenomenal question, and it's something that we're trying to address as well. In the case of Melanogaster and Xanthomea, they will never produce hybrids in the field. Well, never say never, but I would be very surprised if I actually catch a Melanogaster Xanthomea hybrid in the field. Melanogaster does occur in Sao Tome. That is the only island where Xanthomea actually takes place. It's endemic to that volcanic island. At low frequencies in the forest, it does exist. So the possibility of hybridization does exist. In the sense
of trying to find out what are the ecological differences between these species. We have been working trying to find out, for example, about climate niche use and food sources for species that are more closely related, for Yacuba and San Tomé and for Yacuba and Tessieri in the island of Bioko. So it's something that we're definitely interested.
There's another question. So relating to your conclusion that postzygotic isolation might be driven by selection or not, how can we discriminate between this? Do we need to know all the players in the relevant pathways and the relative rates of evolutionary change?
Well, that's a great question. So I think that bold claims require more evidence. So that evidence of natural selection coming into play, causing hybrid inviability, was put forward in the early 2000s by David Presgraves and the work of Daniel Barbach. And in their case, the rate of evolution of the nuclear proteins and HMR is actually quite staggering. It's really, really fast. In our case, we just don't find it. So I think that what I am trying to say here is that there are definitely cases in which positive selection actually does affect, like natural selection at the molecular level drives the evolution of alleles involved in hybrid inviability. That is for sure. We have plenty of evidence of that. But what I am showing you is that there are other cases in which that doesn't happen. Whether we need to know all the players, the reality is that, I mean, HMR remain, like all the components of the HMR that is the best studied incompatibility in Drosophila remain unknown until relatively recently, when Nitin Fatnis actually just went and did a massive mutagenesis screen trying to find out at least the third partner. So it's actually quite hard to find out where all the partners of an incompatibility are. So I don't know if you need to, it depends on how strong your conclusions want to be. In my case, I don't find hybrid inviability in this case, in this phylogenetic group of species, to be driven by selection. But that doesn't mean that it's not. I mean, I just provided a counterexample of natural selection driving always postzygotic isolation. But I am not ready to say what is the relative importance of the process.
Thank you. With regards to examples of reciprocal asymmetries in post-mating interactions or incompatibilities, does this give a signature of variation in rates of evolutionary change and hence differential selection in some lineages?
Oh, that would be, yes. So there is an argument that was made by Roman Jukilevich in the 2012 paper that actually argues that if you have a strong postzygotic isolation in one of the directions of the cross, you will also find, if natural selection is acting, you will also find a concordant level of asymmetry in pre-zygotic isolation. So that's essentially his conclusion. I still think, I am not certain, that if you have certain scenarios of genetic architecture in which pre-mating and post-zygotic isolation are linked and they feed on each other, you could actually get the same level of concordant asymmetries, but I am not really ready to make that assertion.
Thank you. And I think we can end on one more broader question. What do you think are the most important current open questions in speciation research that you think younger generations should think about?
Oh my, that is a big one. I think that one of the reasons, this is a very personal question, so I am going to answer what I think is very interesting. I think that there is plenty of variation in the rate of species formation across the phylogenetic tree of life. And we don't really know how that connects with reproductive isolation. Dan Robowski and I tried to address this question with essentially the power that we had from these comparative studies, but it was a limited time. So essentially, we would like, if possible, to actually just connect macroevolution studies on the rates of speciation with how species formation actually occurs with this kind of microevolutionary information that we can actually provide into it. I think that would be the most exciting thing for me to see.
Very exciting avenues to study further indeed. I think, unfortunately, this is all the time we have for today, but thank you very much again for your excellent
Thank you so much. I really appreciate it. This was a lot of fun.
For everyone else watching, join us again on Wednesday next week when Professor Paul Turner from Yale University will talk about phage selection pressures on bacterial pathogenicity. Until then, have a great week, everyone, and stay safe. Goodbye.